That look good?
That looks great. I will give a couple more seconds just so we start right on time.
Right.
All right, it's 5 o'clock my time. So let's go. First up is Matt Calvert. Take it away, Matt.
Hi, everyone. So my name is Matt Calvert. I'm a PhD student in Corlett Woods Lab here at the University of Pennsylvania. Before I begin, I just want to give a big thank you to the Hamilton Award Committee, to the judges, and also extend a big congratulations to the other finalists. I've just been absolutely blown away by the work I've seen today, and I'm just honored to be presenting alongside of all of you. So my work today is on the fingerprints of host-associated selection in the genome of a hypergeneralist parasite. So host-parasite interactions are a ubiquitous phenomenon on Earth and represent one of the most important drivers of diversity. And the majority of these interactions are specialized, where a single parasite taxa infects a single host taxa. And this generally involves tight coevolution between parasite and host. However, a small but not insignificant number of parasite taxa are generalists, capable of infecting many host species. But due to the ubiquity of specialized interactions, as well as their empirical and theoretical tractability, most work to date on the evolution of parasitism has really focused on these specialized interactions. And by comparison, we know a lot less about how generalists evolved, and in particular, how heterogeneous host selection shapes their evolution. And that really brings up my main research focus of this project, which is to explore the patterns of variation that heterogeneous host selection leaves in the genomes of generalist parasites. So most work that has been done in this area on host selection in generalists has actually been done on species that are pretty much comprised of fairly deeply diverged and highly specialized host-associated populations. An example of this comes from the fly Rhagylides palminella, which is not really a parasite, but still an antagonist. And at the species level, they're capable of using and utilizing a variety of different host fruit species, but actually this species is comprised of different populations which are highly specialized and really only use one of these fruits. And so less is known about how host selection, and I should also say that we know for those more specialized, sort of specialized species, we know a lot about how host selection mediates genomic variation. Less is known about how host selection operates in the genomes of what we would describe as, or what I would describe as a hypergeneralist parasite. So these are parasites that infect dozens to hundreds of host taxa. And some examples of these include Leishmania parasites of mammals, chytrid fungus, which has been devastating amphibian species across the world, and the focus of my talk today, root knot nematodes, which are pretty devastating pests of plants. And so to address this overarching goal of uncovering the fingerprints of host-associated selection in generalists, we had three main research questions. One, simply, is whether the same loci are involved in adaptation to different host species. Two, what form of selection is operating on these host-associated loci, and what can that tell us about the evolutionary process? And three, are host-associated loci enriched for specific function, and what might that reveal to us about how the generalist phenotype? And so we did this work in the northern root knot nematode, which is a generalist parasite of plants. And so root knot nematodes form these gall-like structures on roots that you can see here, and these serve as feeding sites. And so early in life, these nematodes will migrate through the soil looking for a suitable host plant. When they find one, they'll invade the root system, where they'll molt into the sedentary form, where they'll siphon resources away from the host and reproduce. And the species that we work on that I'll be talking about today is Meloidogyne hapla. And so in this plot here, I'm just showing you the number of known host plant families that different nematode parasites of plants infect, or different species infect. And you can see that the great majority of them are fairly specialized, infecting one or just a few host plant families. Our species, Meloidogyne hapla, infects over 80 host plant families, comprising of hundreds and hundreds of host plant species. So to address this goal of thinking about how host selection is shaping variation in generalist genomes, we sampled root knot nematodes in a replicated design across space at 3 sites in Virginia and 1 in Pennsylvania. And we sampled nematodes from the same 3 host plant species at all of these sites, 2 species belonging to Fabaceae, or the legumes, and 1 belonging to Asteraceae. And here is just a look at the number of nematodes that we collected from individual plants belonging to each of these species at each site. And while the results from Pennsylvania are really interesting, I don't have time to go into them today, so I'll be just telling you about the cool stuff we found in these Virginia populations. And so traditionally, there are several barriers to generating sequencing data from field-collected microparasites. And we were able to overcome these by applying technology that was developed for single-cell sequencing, that is, whole genome amplification. And this allowed us to generate sequencing libraries from individual parasites that we collected from the field, which I think is just super cool, and I'm really excited about it. And I think this is a really exciting avenue, new technology for population genetics. And so using these libraries, we have aligned reads to the Maloidogyne haploid reference assembly from Wormbase, and with standard variant calling and filtering, we ended up with about 117,000 SNPs, which may seem like a small number for a whole genome set, but the Maloidogyne haploid genome is very tiny at just 50 megabases, so this is a reasonably sized data set. And so before jumping into the main questions and results, I just wanted to show the results of a PCA analysis that we ran on a set of unlinked putatively neutral SNPs that reveals no evidence for the existence of cryptically specialized host-associated ecotypes like we see in other generalist species. So here, each point is an individual nematode, colors indicate the different sites, and shapes indicate different hosts. And you can see very strong clustering by geography with these distinct clusters by color, but no real clustering by host. So on to our first question about whether the same genetic loci are involved in adaptation or responding to selection from different hosts. Well, the first step we have to do is actually find those host-associated loci. And to do that, we performed a version of a genome environment association analysis. And really what this allows us to do is essentially estimate allele frequency differences between pairs of host-associated nematode populations while controlling for the underlying population structure that exists in our sampling data set. And so here's the result of just one of these comparisons. And on the y-axis, you have this, what we call C2 statistic. It's just the standardized allele frequency difference. And this statistic is scaled, which is why these values are greater than 1. And this red bar just indicates the threshold for significance. And I think it's important here to just take a moment to point out how remarkable it is that we find several regions of host-associated variation when we're sampling nematodes in these sites from host plants that are only a few feet or a few meters away from each other. And we can populate the rest of the slide with the other comparisons and see that there's a substantial amount of host-associated genomic variation and what actually looks like a lot of overlap as well. And we can test this statistically, and we find that host-associated loci, while controlling for non-independence among SNPs that are in linkage disequilibrium with one another, are significantly overlapping across these population contrasts. And this implicates a set of shared genetic loci that could be involved in adaptation to multiple host plant species, but also a fair number of idiosyncratic loci as well. So on to our second question on what form of selection could be acting on these host-associated loci. And here we're really thinking about and looking for signals of positive selection and balancing selection. But how you interpret these signals, if you find them, really kind of depends on the underlying biological process that could be facilitating adaptation. And by that, I mean, if we think that these generalist parasites are actually involved in some kind of coevolutionary interaction with different host taxa, then positive and balancing selection would be indicative of maybe different types of coevolution. If we think actually that maybe there's not so much coevolution happening and rather there's just kind of unidirectional adaptation to different host taxa, then positive selection could be indicative of heterogeneous host selection on conditionally neutral mutations that influence fitness across some or maybe many host taxa, but have maybe no real fitness cost on others, while balancing selection could be indicative of heterogeneous host selection on antagonistically pleiotropic mutations that are beneficial on some hosts but deleterious on others. And so to look for these patterns of positive and balancing selection, we just use the classic statistic Tajima-D. And so low values of this statistic indicate an excess of low frequency polymorphisms, which is often taken as evidence of positive selection, while high values of this statistic are indicative of excesses of intermediate frequency polymorphisms, often taken as evidence of balancing selection. And so we used coalescent simulations derived from a best fitting demographic model and to basically establish statistical thresholds to decide whether loci were under significant positive or balancing selection. And we performed a sliding window analysis where we estimated Tajima's D and non-overlapping windows of 10 kilobases, which roughly approximates the genome-wide average recombination distance. And then we took all of the windows that exhibit a signal of positive selection and asked whether those windows were enriched for host-associated SNPs. So here on the y-axis, we have the proportion of genomic windows that exhibit a signal of selection, and there will be two bars, one colored based off of whether that window contains a host-associated SNP, and one colored for those that do not. And what we see is that genomic windows that exhibit signals of positive selection are enriched for host-associated SNPs, and this pattern is replicated across each of our sampling sites as well. We see a similar pattern for balancing selection too, where genomic windows that exhibit a signal balancing selection are again enriched for host-associated SNPs. And so we can also examine the frequency of these different signals of positive and balancing selection. So here in this violin plot on the left, I have the genomic windows that contain host-associated SNPs, and we can see that similar numbers of these windows, and Tajima's D on the y-axis, and we can see that similar numbers of these windows show evidence of positive selection and balancing selection. So 78 windows showing evidence of balancing selection, 86 windows showing evidence of positive selection. So similar frequencies of these types of selection are putatively acting in our data set. And so I just want to revisit this table that I showed you before. And it really, the results that we find in this sort of equal frequency of positive and balancing selection suggests that each of these are really equally likely. And it really will require some future manipulative studies and maybe some deeper sampling in order to determine how much of these signals that we're seeing are due to actual coevolution between this generalist parasite and its different host taxa, or maybe unidirectional adaptation to different suites of host taxa. And so onto our final set of questions, thinking about whether host-associated loci are enriched for specific functions. So one analysis we did is what we call a sequence ontology analysis, where we basically asked whether host-associated SNPs are enriched for specific genetic elements. And what we find, interestingly, is that host-associated SNPs are way overrepresented in introns. And this is a pattern we're still digging into, but what it seems to potentially suggest is that post-transcriptional modifications, potentially splicing, may play some role in facilitating adaptation to different hosts. But that's something we would need to dive in further and maybe do some manipulative work on. And the final thing that we did, and the final thing I'll present on today, was exploring whether the host-associated variation and signals of selection that we observed overlap with previously identified cross-species eQTL hotspots. So a cross-species eQTL hotspot is a variable region in the parasite genome that is associated with gene expression variation for large numbers of genes in the host's genome, which is why we call them hotspots. And so two of these hotspots are localized to linkage group 8 in the maloidogyne haploid genome. And so here, I'm going to show you contigs that map to these hotspots, as well as contigs that flank these hotspots. And first, I will reveal to you Tajima's D values across these contigs. And you can see that there are several genomic windows that have elevated signals of balancing selection in these eQTL hotspots. And in fact, a genome-wide analysis revealed that windows with signals of balancing selection are actually enriched in these eQTL hotspots. We can also overlay the host-associated SNPs, which you see here with these red circles above the Tajima's D values. And again, we did an overlap analysis and found that these eQTL hotspots are also enriched for host-associated SNPs. And one final thing I'll just say is that 3 of the 5 cross-species eQTL hotspots, including those that I just shown you, mediate the expression of several host plants' MADS-box transcription factors, which are these deeply conserved regulators of plant development. And when I saw this result, I thought it was really fascinating because to me, it suggested potentially, or one idea is just that maybe one thing that supports the maintenance of a generalist phenotype is the ability to manipulate genes that are found across several potential host taxa. And so just to conclude, we found extensive host-associated genomic variation, some of which is implicated in adaptation to more than one host species, that host-associated loci are enriched for signals of positive and balancing selection, and that we need more manipulative studies to figure out if this is from coevolution or maybe unidirectional host adaptation. And then finally, that host-associated loci are enriched in putative regulatory regions and in genomic regions known to mediate the expression of gene expression variation in their host plants. And so with that, I would just like to give a huge shout out to my incredible PI, Corlett Wood, as well as the other co-authors in our lab on this study, as well as my thesis committee and other members in our department. And if there's time, I'm happy to take any questions. And I'll
Great.
stop sharing so that the other person can share.
Perfect. Michelle, you can start anytime, just sharing your screen, not start talking, sorry. And meanwhile, for this whole session, please put your questions in the Q&A. It looks like you have a question in the Q&A, Mac. I'm going to leave you to respond to that, and I'm going to introduce Michelle McCauley, who is our second speaker. Go for it. Thanks.
Okay. Hi, I'm Michelle. I am a PhD student at Emory University with Levi Moran. And I'm going to talk to you about how a co-evolving parasite and increased mutation rate can maintain outcrossing where one alone cannot. So in his 1982 book, The Masterpiece of Nature, Graham Bell said that sex is the queen of evolutionary problems, which is a sort of delightfully dramatic thing to say. So what did he mean by this? Bell meant that sex is a problem because we see it everywhere, even though it is costly. So costs of sex include the costs of finding or attracting mates, costs associated with copulation itself, the cost of meiosis, which is essentially the dilution of the genome across generations, or what I'm going to focus on, which is the twofold cost of males. And the cost of males is the per capita reduction in the number of offspring in biparentally outcrossing populations compared to uniparentally reproducing populations. So if you imagine a population of obligate outcrossers, and one female should gain the ability to reproduce uniparentally, then all else being equal, eventually the uniparentally reproducing descendants of that female will displace the outcrossers just as the sheer numbers game. So if sex is so costly, why do we see it everywhere? And there's a lot of hypotheses to explain the prevalence of sex in nature, and I'm going to briefly go over two. The first one is the Red Queen hypothesis, which states that co-evolving parasites can maintain outcrossing in their hosts, but this hypothesis only functions when parasites are sufficiently virulent as to offset that cost of sex. And this is a problem because highly virulent parasites are not as common in nature as outcrossing is. Another sort of genre of hypotheses have to do with mutation accumulation. And the core of these hypotheses are that outcrossing can prevent a buildup of fixed deleterious mutations within a lineage. But again, there's a limitation. Only sufficiently high mutation rates can maintain outcrossing. And again, super high mutation rates are not as common in nature as outcrossing is. And this is a problem in the fields because we have all of these different hypotheses for sex, but no singular hypothesis can completely explain the prevalence of sex in nature. So every few years, somebody puts out a review that basically says, hey, we have all of these theories for sex. What are we going to do about this? And one possible solution is called the pluralistic approach. And the pluralistic approach basically asks if a combination of parasites and mutation can favor outcrossing at levels where one alone cannot. And I was really interested in this pluralistic approach for my dissertation, and I decided to test it experimentally. And to do this, I basically asked three questions. First, how virulent do parasites need to be to maintain outcrossing in my system? How high do mutation rates need to be to maintain outcrossing? And can combining parasite and mutation lower either parameter's threshold? So to sort of graphically visualize this, I want to parameterize my parasites and I want to parameterize my mutation rates. And then ultimately I want to combine them and see if anything happens here in the middle of my table. So I tested this using a series of evolution experiments using the nematode worm C. elegans. C. elegans are androdioecious, which means worms are either males or hermaphrodites. And they're mixed mating, which means that hermaphrodites can either self-fertilize or they can outcross to males. And I should also note that under standard laboratory conditions, C. elegans are almost entirely self-fertilizing. So all of my evolution experiments sort of take the same form. So pay attention. They're all self-invasion experiments where the goal is to sort of recapitulate the scenario I described before of a single female gaining the ability to reproduce uniparentally. And so we do this with all of our starting populations of worms, our 95% worms that have been genetically modified to be obligate outcrossers, and we add in a small number of worms that are capable of self-fertilization. And these two subpopulations have a shared genetic background. So we take these worms and we expose them to some condition of interest over many generations, and we track their selfing rates over time. So if our condition of interest favors outcrossing, selfing rates will remain low, as in this orange line, and we will say that outcrossing is maintained. But if outcrossing is not favored, selfing will invade the population and selfing rates will increase over time. And as in this blue line, and we're going to call this a selfing invasion. And I do want to note that the rate and degree of selfing invasions can vary a lot. And that in itself is pretty interesting, but for the sake of this talk, I'm just going to say if selfing rates increase, it's a selfing invasion. Okay, so first let's parameterize our parasite virulence. This experiment was done by our collaborator, Sam Slowinski. And so Sam used our worms as the host. And for a parasite, he used the bacteria Serratia marcescens, which conveniently is red to match the Red Queen. And Sam used different strains of Serratia of varying virulence, and he co-passaged this bacteria with the worms in order to allow for coevolution between host and parasite. Okay, so Sam used 4 different strains. Our strains of Serratia varied in virulence are what I've called relatively low, induces less than 10% mortality in our worms. This moderate treatment induces around 40% mortality, all the way up to our very high treatment induces up to 80% mortality in our worms. Okay, so Sam found a successful selfing invasion in his 2 lowest virulent strains, but with his 2 high virulent strains, we saw that outcrossing was maintained across the course of the entire experiment. So Sam was able to find that there's some sort of very clear threshold here within our virulence parameter space. So bringing it back to our table, we can now parameterize our parasite space, and we have 2 treatments which do not maintain outcrossing and 2 that do. So, okay, next we want to parameterize our mutation rates. I did this experiment with the help of my awesome undergrad, Kenya Say. And basically we used the chemical mutagen ethylmethane sulfonate to artificially increase the mutation rates of our worms. We can essentially titrate mutation rate to be whatever we want by varying the concentration of this mutagen. And worms were mutagenized every other generation. So we had 5 different mutation treatments, which varied from our control, which is just the baseline worms mutation rate, very low, 1.5 times the baseline mutation rate, all the way up to a treatment I've called high, which is 8 times the baseline mutation rate. And if that sounds ridiculously high to you, don't worry, it's not. This is still a perfectly biologically relevant mutation rate. Okay, so we found in our 3 lowest mutation rates, we did see a selfing invasion. But with our 2 highest mutation rates, outcrossing was maintained across the course of the entire experiment. So again, we were able to identify that there is some sort of mutation rate threshold within our system. And returning to our table, we now have 3 mutation rates, which did not maintain outcrossing, and 2 that did. Okay, so now that we've parameterized our 2 factors, we can test to see if this pluralistic approach can work. So presumably any factor that by itself could maintain outcrossing, if you combine that with something else, it's going to continue to maintain outcrossing. So for now, we're just not going to worry about these combinations. What we are interested in are these combinations of factors which by themselves could not maintain outcrossing. So we want to see if we can move the threshold for either hypothesis with these combinations. So to do this, I essentially just combined the protocols of these 2 previous experiments, mutagenizing worms and exposing them to parasites. For these results, the axes are exactly the same as before, but the panels represent 3 different parasite treatments. The left panel is our avirulent control, which is just heat-killed Serratia. So this panel will essentially just be the mutation by itself. Our middle panel is that lowest virulent Serratia treatment. And our right panel is that moderate virulent Serratia treatment. And the lines within each panel are our different mutation treatments. And I've retained the color coding from before. So first, let's look at our mutation control. So these lines are essentially parasite only, and we see selfing invade across the board. Now looking at this left panel, our avirulent control, these lines are essentially mutation treatments only. And again, we see selfing invade across the board. This is really good news. It means our controls worked. Okay, so now we can get into our combination of low virulent parasite plus low mutation rates. And so we finally start to see something interesting happen. In this combination of our low virulent parasite plus our low mutation treatment, we see that the selfing does invade, but the invasion is slowed by a lot. We don't see an increase from the starting rate until generation 24. So then moving to our combination of our moderate parasite and mutation, we finally see a combination of treatments that are able to maintain outcrossing across the entire experiment. And this is really exciting because the idea of this pluralistic approach has been around for about 30 years now, but there's very, very little empirical testing for it. So this is some of the first evidence that this pluralistic approach can function in a real living system. All right, so bringing it back to our table, we found one combination which did slow a selfing invasion, and we found one combination that was able to completely maintain outcrossing across the experiment. And so by showing that a combination of factors can maintain outcrossing where one alone cannot, we open up this parameter space under which our various hypotheses for sex can function, which means we're one step towards explaining the prevalence of sex in nature. So as some sort of concluding thoughts, organisms live in complex environments and must contend with many complex selective pressures and forces. So there's no reason to think that only one factor should be contributing to the maintenance of outcrossing. And so by broadening the parameter space where outcrossing can be maintained, we've sort of opened up the floor to more natural scenarios which could maintain outcrossing. And so with that, I'd like to thank my lab. especially my wonderful undergrad and second pair of hands, Kenya, our collaborators, our funding sources, the organizers and judges, and all of my fellow presenters for such awesome talks so far today. I've had a great time. And with that, I'd be happy to take any questions.
Thanks, Michelle. As always, put your questions in the Q&A. Chris, you can go ahead and put your slides up. Great, thanks. Okay, our third speaker in the session is Chris Blake. Take it away.
Thank you. Good morning, everyone. I'm Chris. Thank you for the great opportunity to present my work here today. I would like to start by acknowledging the traditional custodians of the land where I'm conducting my research here in Melbourne, the people of the Kulin Nations, and I pay my respects to the elders past, present, and emerging. Now, when I tell somebody that I'm studying evolution, often people think I'm trying to prove that it's true, and it happened at some time in the past. However, we all know that studying evolution doesn't only mean we are trying to understand what happened in the past. It also means we're trying to predict how evolution might shape the future. And this is key to tackle global challenges such as climate change and the loss of biodiversity. Now, this not only applies to plants and animals, but it's very important for microbial communities as well. And microbes are interesting in a way that they actually can evolve so rapidly that our idea of a boundary between ecology and evolution is starting to disappear. And these processes can occur on the same time scale and therefore directly influence each other's trajectories, leading to eco-evolutionary feedbacks. So today, I want to give you an example of how evolution can shape a global ecological phenomenon and what we should consider when we want to apply evolutionary principles in order to tackle the challenges that come with it, such as the loss of biodiversity. So the phenomenon I'm talking about here are tipping points. And I guess most of you have heard of the term, especially when we think of climate change. A lot of our ecosystems, such as coral reefs or freshwater lakes, are at risk of a collapse because they are predicted to approach one of these tipping points. But tipping points are actually much more common, and we also can find them on a small and localized scale, such as in our gut microbiome or in a soil microbial community. And basically, it's always the same. Tipping points are this critical threshold where even a small change in the environment can lead to a rapid shift in the state of our ecosystem, often changing the community composition, its functionality, and leading to a loss of biodiversity. Now, I would like to use the example of the lake ecosystem to walk you a bit more through in detail what the key ingredients of a tipping point are, because I think it's maybe the most relatable of these examples, but it applies to all the others as well. So here you can see a graphical representation of a tipping point. Basically, on the x-axis, we are mapping an environmental condition, such as the influx of nutrients, and on the y-axis, we are mapping the state of our ecosystem. So in this very simplified example, our lake can take on two different stable states. First, state A, which would, from our point of view, be the desired state, is a very pristine lake. It's very clear. It's dominated by macrophytes that inhibit phytoplankton. On the other side, state B is a very turbid lake where phytoplankton dominate and inhibit macrophytes. Now, where it's getting interesting are the bits in between here, where actually both of these states can be a stable outcome, and where the lake will end up depends on where it's starting. So if we start above our threshold line, That means there are enough macrophytes in the system to keep the phytoplankton in check, and therefore it will always converge on state A, the clear lake. However, once we cross the threshold and go past the tipping point, what happens is there are more phytoplankton in the system now. Therefore, the water will get more turbid. There will be less light for the macrophytes. They will perform worse and then release the competitive pressure on the phytoplankton. They in turn can grow more, the water gets even more turbid, there will be less light, until this positive feedback loop here drives our system all the way down to state B. So basically, we can find every possible combination of an environmental condition and ecosystem state to map out the whole space and identify under which conditions our lake would end up in state A, here in green, where would it end up in state B, here in gray, and then identify the tipping point in orange in between. Now, this is a very well-established theory in ecology. However, until now, we mostly treat tipping points as very fixed critical thresholds, and we don't really know how evolution could change and shape them. So there's a few possible outcomes we could imagine. Evolution could increase the stability of a community, increasing its resilience against an environmental stress. But we could also imagine it could destabilize a system, like in the forms of an arms race of two species competing for each other. So in our lab, we set out to develop an experimental system to study how evolution can shape tipping points. And we ended up using a co-culture of E. coli and yeast. And I think this is a pretty cool system because both of these organisms are model organisms in microbiology. They are well understood, but we wouldn't think of them to occur in the same environment, and therefore we don't expect them to have a shared evolutionary history. So now we wanted to look at how evolution could shape a tipping point of this culture from two different angles. The first was coevolution. So basically here in the system, we don't have an external selective pressure other than the two species having to coexist for a long time together. So basically what we did here was we put them together in a culture and then passage them every day for 400 consecutive days, giving us 4,000 generations of evolution. Now, the second angle I wanted to look at was what happens if our culture can evolve to resist an acute environmental stress. For this condition, I chose the antibiotic tetracycline, which is a bacteriostatic, meaning it's arresting E. coli's growth. It doesn't have any effect on yeast. So basically, we can very precisely manipulate growth conditions for E. coli. So in this experiment, I chose the ancestral co-culture, which does not have any shared evolutionary history, as well as the one which evolved for 4,000 generations together, grew it in concentrations of tetracycline for a few days, and then re-isolated resistant pairs. So ending up with these 5 co-cultures now, which all have a different evolutionary background, I wanted to assess the stability of the community and find the tipping point of all of these cultures. For this, I'm mixing all of them in different initial ratios, starting everything from 95% E. coli all the way down to 5% E. coli, basically mapping all the y-axis on this plot here. Then I grow them in a different range of tetracycline concentrations to map out the whole x-axis. And finally, to assess how frequencies change over time, I'm using flow cytometry. And basically, because E. coli and yeast cells are so different in their size and shape, we can easily distinguish the two of them using flow cytometry and get high resolution counts. So I just want to quickly show you how my data then looks like. So here you can see on the y-axis the frequency of E. coli, meaning at a frequency of 1, E. coli went to fixation and outcompeted yeast. At the frequency of 0, it went extinct. And as you can see, over a period of 5 days, no matter at what frequency they are starting at, they are always converging to somewhat of an equilibrium after just a couple of days. Now the picture starts to change slightly when we introduce tetracycline into our system. So here you can see the ancestral co-culture, which is sensitive to tetracycline, under an intermediate stress. And if we look closely here, the cultures where E. coli is starting at very low frequencies, they're going extinct after just a couple of days. So these frequencies I will map as extinction in my plot. On the other hand, if E. coli is starting at higher frequencies, they will remain in co-culture for the whole period of the experiment, meaning everything above will be mapped as coexistence. And then finally, the frequencies, the area here in between, where it's uncertain and it could go either way, I will map as a tipping point. Then, by repeating this experiment over and over again, increasing the concentrations of tetracycline, we can basically plot this slice after slice to get to our final tipping point maps. Now, I would like to start with the good news. What happens if a system is able to evolve towards an acute environmental stress? We see, if we compare these now, the space where they can coexist is increasing immensely, and the tipping point is delayed and pushed out all the way to the right. However, there's also a downside to this. If we have a look at all three of the co-evolved cultures, we can see a second gray area appearing, a second tipping point, where under low concentrations of tetracycline, E. coli is actually able to outcompete yeast. So this experiment clearly shows that tipping points are not fixed thresholds, but evolution changes them. And it can do so in both ways, either delaying or facilitating an earlier collapse. Now, because a lot of things can happen during 4,000 generations of evolution, we wanted to get an idea of which traits are actually driving the system, especially if you want to think of enhancing the stability of a community. Which of the traits should we focus on? Is it growth conditions, or is it the interactions between the two species? So for this, we modified the classic Lotka-Volterra two-species competition model, basically to include this positive feedback loop I introduced earlier in my talk. So for this, we changed the two competition coefficients, so the effect yeast has on E. coli, as well as E. coli on yeast, to be positive frequency dependent, as seen with the phytoplankton and the macrophytes. Now, when we use this model and parameterize it with the ancestral data we are getting, we're actually able to recapture a tipping point, which looks very, very similar to our observed data. Now we can use this model to change each parameter individually to see how a change, an evolutionary change in it, could influence the tipping point. So in this example here, you can see what happens if competition between the two species is increasing. That's actually where we see a second tipping point occurring, and our system will be destabilized. On the other hand, if we are decreasing the competition coefficients between the two species, that's already enough to stabilize our system and delay a collapse and push out the tipping point. And finally, probably the best strategy would be if we can evolve resistance towards the environmental stress without changing any of the other parameters. So in conclusion, what I want you to take home from this talk is currently we are thinking of tipping points as this fixed threshold, but they are definitely not fixed. And evolution changes and shapes them. And it can do so in both ways. Evolution can stabilize a community and delay its collapse, but it can also destabilize a community, leading to an earlier collapse. And with this, I would like to thank you all for listening and thank the organizers for this great opportunity. And very big thanks to my lab group, the McDonald Lab here in Melbourne. Thank you.
Great, thanks. As always, put your questions in the Q&A. Nitin, you can go ahead and put your slides up. We do have a minute or two.
Do you want me to wait till 4:45?
I think so, yeah. We'll just give it another minute. And we stay on track, roughly.
Okay.
Thanks. Okay, I think it's that time. Next, we have Nitin Ravikantachari, and take it away. Slides look good.
Thank you so much. Hello, everyone. Thanks to Evolution for organizing this conference and this opportunity. And it's been great hearing all the amazing talks that's been happening so far. I am Nitin, and today I'll be talking about genetic mechanisms underlying preference performance mismatches. And before I get into my study, I'd like to talk about local adaptation. Heterogeneous environments lead to locally adapted populations. So as you see here, due to differences in local environments, the mean fitness of a population, for example, the population in South, is higher in its native site compared to any other foreign site. And local adaptation is maintained through the interplay among various evolutionary processes, including drift, selection, and gene flow. And each point here in this graph that I'm showing on the right and left represents the difference in fitness between sympatry and allopatry averaged over the whole metapopulation. So, assuming strong selection in a temporally stable environment, we would expect high rates of local adaptation when gene flow is low. This is because high rates of gene flow can lead to maladaptive gene flow occurring into the habitat or into the population, thus destabilizing whatever local adaptation is occurring. However, in a temporarily fluctuating environment, intermediate levels of gene flow increases local adaptation. And this is because it helps in increasing standing genetic variation, thus helping populations respond to changing environmental conditions. However, novel environments can derail locally adaptive populations, especially if there is rapid environmental change. So today I'll be talking about one such type of a novel environmental change known as evolutionary trap. So if we plot the fitness value of a resource against the preference for that particular resource, we would expect locally adaptive responses to fall along this yellow line. So what we expect is that when the preference for a native resource should increase with increase in its fitness value for that particular resource. However, in the presence of a novel resource, it can lead to one of two novel interactions highlighted in brown circle here. If the fitness value of a resource is high, but the preference is low for that novel resource, it goes unused as an undervalued resource. However, if the fitness value of the novel resource is low and the preference for that novel resource is really high, it can lead to preference performance mismatches, or what we call evolutionary traps. And today I'll be discussing one such scenario between a native herbivore and a novel host plant that it interacts with. So the native herbivore that I'm interested in is known as Pyrrhus mactanoi. It's a butterfly that's distributed in the Rocky Mountains. And these butterflies are specialist herbivores, and they feed exclusively on Brassicaceae, that is the mustard family plants, because they have a specialized enzyme known as nitrile specifier protein that can help them detoxify the mustard oils or the glucosinolates that is present in the plants. And the females of this butterfly, ovipositor and the larvae, lay eggs on a variety of native plants, and they use glucosinolates as cues to find the plants to lay eggs on. And a novel invasive plant that I'm interested in is called Claspia arvense. It's a mustard plant. It's a mustard plant. It is invasive to the US, but it is native to temperate Eurasia. It is distributed patchily in the landscape, and this plant mainly contains one type of glucosinolate or mustard oil known as synechron. So what is the evolutionary trap? So the females of this butterfly recognize class P as a host plant because it shares a lot of Q similarity with the native host plants. And the Q similarity is synechron, the glucosinolate. And when females lay eggs on the plant, most larvae die before pupation, thus leading to an evolutionary trap. However, we do know that there is variation in time to death in the larvae when they feed on the toxic plant, and they can be rescued if they're provided with native hosts. So I'll be addressing two questions today. The first one is, are there any signatures of local adaptation in areas where the butterfly and the plant co-occur? And the selection act on larval ability to feed on the toxic plant or on the female's ability to distinguish between the plants. So going to the first question, if there's any, to understand if there are any signatures of local adaptation, what we did was we did a whole genome sequencing of about 335 individuals in the East River Valley in Colorado. And we had about 140 individuals from areas where the plant occurred, and about 195 individuals from areas where the plant did not occur. And the three regions colored in yellow are the regions where both the native plant and the invasive plant occur. And the eight sites here colored in blue are areas where only the native plant occurs and there's no invasive plant. So the first thing we wanted to see was that we wanted to see if there was any population structure in the butterflies among these sites. So we found no evidence for population structure in the butterflies. Our PCA results as well as our admixture results indicated that the entire valley was comprised of a single population. We then tested if there were any signs of genetic differentiation among sites. So we used FST measures to do that. And our pairwise FST comparisons among all sites revealed that there's absolutely no genetic differentiation. And we also used population scale allele frequency covariance matrix to see if how the sites were correlated with each other. And what we found was that all sites were positively correlated with each other, saying that, basically indicating that they're all one and the same and there's no genetic differentiation. So given that, we wanted to see, can selection occur, or there are any signs of selections even in the presence of no genetic differentiation or no population structure? So what we did was we looked at XTX measure, which is here on the y-axis. So it's a measure of adaptive differentiation. So we looked if there were any loci under adaptive differentiation, and we saw about 8,600 loci that were under adaptive differentiation. And we also looked at genotype by environment association using a base factor, so 100:1 odds of being associated with that particular trait and that particular environment, and that particular environment being the presence of class P. And we found about 1,008 loci that were associated with the presence of class P. But what I was specifically interested in is to find out if there were loci that were both adaptively differentiated as shown by the XTX scores, as well as those and associated with class P RNC as indicated by BF values. And we found about 9 loci that were both adaptively differentiated as well as associated with class P RNC. And when we looked at what these loci were, we found that most of the loci were associated with development and metabolism. So this led to our next question to see or to ask, where is selection occurring? Is it occurring on larval ability to feed on the toxic plant or on the female's ability to distinguish between the plants? So to do that, what we did was we conducted a female oviposition choice experiment, a simultaneous female oviposition choice experiment, where we got the females, we gave them a choice between the native and the invasive plant. We collected their eggs, quantified how many eggs they laid on each plant, and then we quantified preference, and we did RNA-seq on the female tissues. And to look at larval performance, we gave third instar larvae after starving them for about a 24-hour period, either the native plant or the toxic plant, and we let them feed for 24 hours. We made sure that the gut were full with the plant, and then we degutted the larvae, and then we did RNA-seq on that. And this is what we found. So we did not find any differential gene expression in the females that preferred one plant over the other, whereas we found a lot of genes that were differentially expressed in the larvae that fed on the native plant versus the toxic plant. So this kind of, this kind of pointed us to the direction that selection could be acting on larval ability, but we wanted to explore further. So what we did was we looked at what were these genes that were differentially expressed and we wanted to see the mechanistic underlyings of it. So we identified 3 distinct gene clusters that were differentially expressed between the larvae that fed on the invasive and the native plant. So green here is the invasive plant and brown is the native plant. And our hierarchical clustering of larval transcriptome profiles indicated that larval transcriptomes followed host plant specific treatment patterns. And however, there were 2 larvae that fed on the invasive plant that showed a profile similar to the native plant, suggesting that there is a lot of variation in how the larvae are able to cope up with the toxic plant. So with the 3 gene clusters, the first gene cluster, in the first gene cluster, we saw genes that were upregulated in larvae that were feeding on the native plant. And these were all related to, and the gene ontology terms that were related to genes in this cluster were related to favorable host plant feeding, including lipid metabolism, reproduction, regulation of transcription and translation, and breaking down of complex carbohydrates. And when we looked at the cluster 2 and cluster 3, which were upregulated in larvae feeding on the invasive plant, we saw gene ontology terms that were enriched for glucose metabolism, which is breaking down simple sugars, maybe due to not able to break down the plant compounds, response to stress. They were exhibiting gene signatures of cell death and apoptosis, and they were also showing accelerated development. So we were then interested in looking at what could be causing these differences. And one of the things that we wanted to check was the differences in gene expression profiles of the detoxification genes. So when we look at insect detoxification genes, there are 3 phases. So the first phase is functionalization, which is oxidoreductase activity. And this is where P450s and coesterases occur. And these are employed by generalist herbivores, and the phase 1 enzymes act on a variety of substances to detoxify them. And phase 2 is where conjugation occurs, where specific functional groups are added to the toxin to make them water soluble. And this is where the nitrile specifier protein occurs. And the nitrile specifier protein is where we see most of the brassicaceae feeding butterflies detoxifying the compound present in the mustard plants. And phase 3 is excretion, and phase 4 is general digestive efficiency. So what we saw was that, so the graphs here represent the log CPM counts per million of the expression values, and phase 1, phase 2, phase 3, and general digestive enzymes responding to each of the enzymes that are present there. What we see is that phase 2 enzymes, which is the nitrile specifier protein, is only expressed or enriched in the native larvae. And phase 1 and phase 3 are enriched in the invasive or in the class P arbency feeding larvae. So this suggests that the larvae are unable to effectively use the nitrile specifier protein to detoxify the compounds that are present in the invasive plant. So to summarize what we found so far is that we found no genetic differentiation among any of the butterflies across the valley between the areas where the plant co-occurred or the plant where the plant and the butterfly did not occur. Yet we found signatures of strong selection in areas where the plant was present, and we found that selection was, we found differences in gene expression only in the larval profiles, but not in the female oviposition behaviors. And when we looked at what these expression profiles reveal, we found that the larvae were unable to effectively use the nitrile specifier proteins or the phase 2 enzymes to effectively detoxify the chemical compounds present in the invasive plant. So what does this imply to understand preference performance mismatches or the evolution of host plant interactions on novel host plants? So we know, so from this, what we can say is that the deep coupling of oviposition preference and larval performance can lead to evolutionary traps, and we can find the evolution, or we can find evidence for local adaptation even in the presence of high gene flow and low genetic variation. And likely escape from this preference performance mismatches would be the larval for the larval ability to feed on the plant rather than females' ability to avoid laying eggs on the plant. And our study actually provides more mechanistic understanding of for error-prone oviposition as a fuel for evolution of host plant range in herbivorous insects, as repeated error-prone oviposition can lead to larvae feeding on new host plants. So with that, I'd like to acknowledge everyone who's helped me with this, including my committee members and people who help collect the data. And I'll be happy to take any questions if there's any time.
Great. Given the time, we're going to move on. Our final speaker of Hamilton 2024 is Mackenzie Urquhart Cronish. And you can go ahead and share your slides.
Okay, thank you.
Thank you.
Okay. So a major question in evolutionary biology is what generates and maintains the incredible reproductive diversity we see between and within flowering plant species today. So this diversity ranges from plants with separate sexes to individuals with self-compatible flowers, plants with a diverse range of pollinators facilitating reproduction to no pollinators needed, and the maintenance of asexual reproduction, including clonal vegetative growth. Oops. So an explanation for the maintenance of reproductive diversity within species includes the reproductive assurance hypothesis, where selection favors the ability to self-fertilize or reproduce clonally in habitats where mates and/or pollinators are scarce. The reproductive assurance hypothesis often focuses on how contemporary ecology, like pollinator or conspecific densities, can drive these spatial patterns in plant reproductive diversity. However, spatial patterns in reproductive assurance can also be explained by historical processes, such as post-glacial range expansion. And this idea stems from a classic observation by Herbert Baker, where successful colonization of a novel habitat is facilitated by the ability to self-fertilize and/or reproduce clonally. While I acknowledge that these two hypotheses are not mutually exclusive, contemporary ecological explanations for spatial variation and reproductive assurance have not always been supported in the literature, and the role of historical range expansion in shaping these patterns in reproductive assurance is relatively understudied. So I'll illustrate in more detail how these demographic changes underlying serial founder events during range expansions could not only structure gradients in population density, but spatial patterns in reproductive assurance within a plant species. So in this example, we have individuals who are outcrossing in pink, and those capable of reproductive assurance, such as autonomous selfing or clonal propagation in blue. And in this high-density range core, outcrossing individuals are expected to have a relative fitness advantage over self-compatible individuals due to the avoidance of inbreeding depression. Oops. However, during range expansion, if an individual incapable of reproductive assurance colonizes new habitat, it will not successfully establish since it cannot successfully reproduce on its own. If an individual capable of autonomous selfing or clonal growth colonizes this new habitat, it will be able to successfully establish, and individuals with this trait will be more likely to successfully colonize and establish future populations. And this will lead to a spatial gradient in increased reproductive assurance from that high-density range core to the low-density range edge. And so this leads me to my hypothesis for my project, that serial founder events characterizing range expansions drive the selection for reproductive assurance at the range edge. And given my hypothesis, I predict that autonomous selfing and propensity for clonal growth will increase along pathways of historical range expansion, in line with Baker's law, where we would expect an enrichment for the capacity for uniparental reproduction, meaning selfing and clonal growth following colonization. So an alternative hypothesis, which I did not have the capacity to concurrently test, would include the role of contemporary ecology in shaping these patterns of reproductive assurance. So to test my hypothesis and predictions, I used Erythranthe lewisii, or northern pink monkey flower. It's formerly the northern subspecies of Mimulus lewisii, as my focal species. So Erythranthe lewisii is native to alpine riparian habitat in northwestern North America. It's a perennial species, it's diploid, and it's thought to be typically outcrossing, but it's capable of autonomous self-fertilization and clonal growth via rhizomes. So the map here depicts occurrence records of lewisii across its entire geographic range, and the white overlay on the map is the last glacial maxima of the continental ice sheet. So this project has two components. One is a greenhouse common garden experiment to measure range-wide variation in sexual and asexual reproductive assurance. And the second part is phylogeographic inference using genomic data to provide the relevant spatial context to interpret the influence of historical range expansion on patterns in reproductive assurance. To complete this project, I did a lot of field work. I conducted range-wide sampling over 3 field seasons to collect seed and leaf tissue samples. The map here shows the same range-wide occurrence records. They're now in gray, and the larger points are populations that I sampled. Pink points are places where we sampled seed and leaf tissue, and green points are places where only leaf tissue was sampled. Circles represent sites we visited in person, and triangles are herbarium specimens we used to supplement our data set from that difficult to access northwestern range edge. And of the sampling locations represented by circles, I personally visited 30 out of 31 sites, so everywhere except Olympic National Park. I did not do this fieldwork alone. I have many people to thank, especially my wonderful field assistants, Ken, Olivia, Francisco, and Julian. So to generate my genomic data set, I extracted DNA from leaf tissue samples and completed ddRAD-seq library prep at the University of Montana Genomics Corps before sending my libraries off for Illumina sequencing. After filtering the data, I retained 356 individuals with mean site depth coverage of between 10 and 55 reads in my final data set. And I used this data set to examine range-wide population structure and conduct phylogeographic analyses, which I will give more detail on later. So for measures of reproductive assurance, I conducted a greenhouse common garden experiment where I grew plants from 30 different populations, which are all these pink points on the map that's inset that span the entire geographic range of lewisii. And I grew these from seed to flowering in a pollinator-free environment using pollinator exclosures that I built using a PVC pipe and a lot of bridle tool pictured here. For measures of autonomous selfing, I marked focal buds before flowers opened, monitored fruit development, and collected fruit samples after dehiscence. I repeated this across 3 different buds per individual plant. To quantify clonal growth, after the final self-seed samples were collected, we harvested biomass and counted the number of rhizomes each focal plant produced. And to quantify autonomous seed production, I counted seeds by hand and weighed the samples. So it should be noted that both rhizome and seed count data sets were highly zero inflated. So we also focused on the probability of producing self-seed and rhizomes as measures of reproductive assurance. So first, we wanted to establish whether spatial variation in reproductive assurance existed range-wide in lewisii, and this is in the absence of information about historical range expansion. We detected significant geographic variation in the probability of producing self-seed, and this is indicated by a log likelihood ratio test that demonstrated a better model fit when population was included in the model as a random effect, as well as significant variation in the probability of producing rhizomes, which was also indicated by the same log likelihood ratio test. Given this, next we asked whether this spatial variation in reproductive assurance was related to historic pathways of range expansion. So for our phylogeographic analysis, our first step was to summarize population genetic structure to identify whether there was evidence of multiple refugia contributing to historical range expansion. So here, this figure shows a principal component analysis depicting the overall genetic similarity of individuals in our data set. So looking at the PC plot, we identified 3 potential distinct genetic clusters with 2 putative hybrid clusters along PC1 and PC2. Given this, we took an ancestral cluster number of 3 and input that into an admixture analysis. So here are the results of our K=3 admixture analysis with individuals and sampled populations organized by longitude. We detected all individuals within 2 discrete populations as having assigned hybrid ancestry, and individuals in these 2 populations mapped discretely with these 2 putative hybrid clusters we originally identified in our PC plot. And given their hybrid ancestry, we excluded these individuals within these clusters from further downstream analyses. So here I'm showing the majority ancestry from the admixture analysis from all other individuals mapped to our PC plot. And generally, we see a discrete clustering of ancestry assignments in PC space corresponding to those 3 ancestral clusters. We then estimated genetic differentiation among our 3 major clusters and found evidence for strong differentiation among clusters. And finally, we mapped average proportion of ancestry assignments in geographic space and found strong geographic structure in addition to our strong population genetic structure. And together, these results suggested 3 distinct ancestral clusters, indicating 3 historical glacial refugia. So in the context of our population structure results, we used our genomic data to infer the direction of range expansion between populations within each ancestral cluster. So to do this, we use the directionality index statistic psi, which is a method that describes asymmetries in allele frequencies that arise during range expansion, when serial founder events create a pattern of isolation by distance and genetic drift increases in populations towards the range edge. We then combine this genomic data with the spatial location of all of our samples in the time difference of arrival algorithm to identify the most likely location for the origin of range expansion within each ancestral cluster. And so the TDOA approach identifies the most likely origin of expansion by locating the DEAM that explains the highest proportion of variation in the correlation between these pairwise differences in the size statistic and our geographic distances. So the results of our phylogeographic analyses identified 3 putative refugia, the locations of which were also supported by a comparative phylogeographic meta-analysis of glacial refugia in northwestern North America. And additionally, our other estimates of genetic diversity were, on average, generally highest at mid-latitudes, which also supports these identities of putative refugia. So now, with the resolved spatial locations of our inferred refugia, we're ready to test our central hypothesis and predictions. Does reproductive assurance increase on average along pathways of historical range expansion? To orient you all, the x-axis for both of these figures is the topographic distance between populations and their inferred refugia. The y-axis is the probability of reproductive assurance, either in producing self-seed or rhizomes, with 0 being never producing and 1 being always producing. And these results that I'm showing are the average model outputs of 0 inflated models, where I controlled for ancestral cluster and repeated sampling among individual plants. Okay, so we find evidence that the probability of producing self-seed increases on average with increasing distance from refugia, with the effect size approximately doubling from the historical core to the range edge. And for measures of asexual reproductive assurance, we also find evidence in the probability of producing rhizomes increases on average with increasing distance from the refugia. And together, these results are consistent with hypotheses underlying Baker's law and support the role of historical range expansion driving contemporary spatial patterns in both sexual and asexual reproduction in Lewisiae. So future directions can include tests of how population-level inbreeding depression versus drift load changes in the context of range expansion, given our detected patterns of increased probability of selfing and clonal propagation towards range edges. And also to gain a complete picture of the drivers of reproductive assurance in our system, future work should investigate the relative role of contemporary ecology in contributing to these spatial patterns. So in closing, we tested and demonstrated support for Baker's law, a classic and long-standing concept in evolutionary biology. Generating the phylogeographic data sets necessary to test the effect of post-glacial range expansion on plant mating system evolution has been difficult in the past, since high-caliber tests require data on both reproductive assurance and phylogeographic reconstructions. But with the increasing accessibility of large genomic data sets that include range-wide sampling, we're now, as a field, poised to effectively address these questions. So our study joins a handful of previously published work that tested and provided support for historical range expansion that shapes spatial variation in autonomous selfing ability. But a unique contribution from our study, and to our knowledge, the only test so far, is the addition of asexual reproductive assurance. So in addition to autonomous selfing ability, we also detected spatial patterns in clonal propagation related to historical range expansion. And this aspect of reproductive assurance, clonal reproduction, is generally understudied relative to its sexual counterpart. So given these emerging results, we suggest post-glacial range expansion may be a general mechanism shaping geographic variation in plant reproductive diversity, and future investigations across a diversity of plant species will further inform our understanding of its generality. So with that, I'd like to acknowledge my supervisor, many collaborators who helped with fieldwork, lab work, sequencing, the greenhouse experiment, genomic analyses, and my sources of funding. And with that, thank you all for listening, and I'd be happy to take any questions.
Great.
All
Put
right.
any questions in the Q&A, please?
Could stop sharing as well. That would be-
Sure. Though the hot pink is nice.
It is nice. Yeah. It was the pink was popping at like peak greenhouse experiment.
Yeah. It's a lot of pink. Okay. Well, that wraps up. Oh, comment. Yes, from Kurt Lively in the chat. That wraps up Hamilton 2024. Thank you all. We had an amazing set of talks, 15 today. And it's really exciting to see all the fresh faces doing really exciting evolution work. So thank you, everyone. Congrats to all the finalists.
Mackenzie, did you, sorry, I'm just seeing the question in the Q&A, if that's,
Yes.
we'll leave it open and so you get a chance to answer that question if you want.
Oh no, the question, yeah, I will.